And today we're going to have our fourth and final speaker, Gustavo Ruiz-Pulido. So thank you for joining us. Gustavo is a PhD student in the Tecnologico De Monterrey, Mexico City. Gustavo earned his bachelor's degree from the Tecnologico De Monterrey in Mexico City. He moved to London to pursue his master's degree in Biopharmaceuticals at King College London. And the title of Gustavo's talk today is Tribo-rheological analysis of reconstituted gastrointestinal porcine mucus under different pH conditions. Welcome, Gustavo.
Hello, hi everyone. Thank you for joining this presentation. Actually, as Doug mentioned, I will talk about the Tribo-rheological analysis of reconstituted gastrointestinal porcine mucus under different pH conditions. Actually, it's important to mention first that mucus is a biological barrier that covers every wet epithelial surface in our body, so it's quite important. As well, the mucus is a natural hydrogel that is formed by the entanglement of mucins. Mucin is an important protein because actually it gives the mucus their viscoelastic, their structural, and some of their functional properties. So it's important to study it. As well, I want to mention that the mucus protect us from viruses, bacteria, external particles, but also it is a barrier for the drug delivery or some molecules that we want to deliver. So the objective in this research is to characterize in a better way the mucus, actually using Tribo and rheology in order to understand more about these mechanical properties of mucus and also how the pH in the gastrointestinal tract change the mucus properties, because I want in the future to talk about more drug delivery. The results that I have obtained in terms of rheology, we can see in figure 3, there is a graph in which I plot the modulus, the elastic and the viscous modulus of samples. I performed the experiment under 4 different pH. There are the more common pH in the gastrointestinal tract, the pH 2, that it's very acidic in the stomach. pH 4 is the transitional pH, and pH 6 and 7 that are more related with the intestines. As we can see with the black dots that are related with the acidic conditions, there are anelastic dominant properties. It means that under the stomach, mucus acts more like a gel. It's a protective gel because the harsh acidic conditions could start to auto-digesting our stomach. So that's why it's more like a gel. But in the neutral pH, probably 6, 7, with the blue and green figures, we can see that the modulus are quite similar. Even there are very slightly crossovers, which means that the sample is more, it performs more like a viscous polymeric solution. So it's more like lubricant properties than protective properties. As well, in figure 5, we can see that independently of the pH, the viscosity of all the samples is reduced when the shear rate is increasing. It means that mucus acts as a lubricant, even if it's on a gel or it's on a solution. Of course, on a solution is a better lubricant, but at the end, is lubricant under all conditions. Finally, I perform a tribological analysis through a Stribek curve in order to know how the coefficient of friction interacts with the mucus. As it was expected, the pH 2 or the acidic condition exhibit a higher coefficient of friction because it acts more like a gel. The function of mucus here is more to protect. But on pH 6 and 7, the mucus exhibit the lowest coefficient of friction because it's more lubricant. That's why it's a very good thing to understand these kind of conditions. The relevance and the future perspective of this project, first of all, the relevance is because studying the viscosity and the coefficient of friction under condition that mimics the human body conditions, it's important to understand more about this functional barrier because it's quite important to know all the properties. Because in the future, as I mentioned, the perspective is to develop some nanoparticles for drug delivery. And one important interaction between nanoparticles and this mucus is the mucoadhesivity, which can be evaluated through Tribo-rheology, but also can tell us if the nanoparticles are modifying the structure or the properties that should be very important, for example, the protective and the lubricant. So that's why I am focused now on characterizing mucus in order to pass in the future for the interaction with these nanoparticles. Thank you for your attention, and I want to acknowledge Medina's group, Tecnologico De Monterrey, CONACYT, the Mexican Council of Science and Technology, and also the AOCS annual meeting and Expo. Thank you.
Thank you, Gustavo. A wonderful presentation. Something I didn't realize about mucus. So, it certainly is a very important barrier and conduit to transmission of things into our body and keeping bad things out. So, great talk. Judges, we have some questions for Gustavo.
Yes, so thanks a lot, Gustavo. Very interesting topic. Can you please elaborate more about implication and a possible application also of your research?
Well, thank you for the question. Actually, it's quite interesting because the application of this research, first of all, is to know more about the properties that has this biological barrier in terms of the protection and the lubrication that are one of the main characteristics, because mucus has an isoelectric point around 2.6, 2.7. So that's why it changed their structure from a gel to a solution. That's the first and important thing to mention here. And also because, as I mentioned, in the future, I want to start to performing some experiments with different polymeric nanoparticles, because there is a recent interaction that has been studied that is called mucoadhesion or bioadhesion with mucus, in which the nanoparticles could interact through different mechanisms with the mucus, but this interaction sometimes could affect this change, this structural change. And it's important to keep these changes because, well, as the mucus is a protection. So if, for example, in the stomach, it acts more like a solution, probably it could affect our digestion because the acids could auto-digest the stomach. It's a very extremely example, but it's something to take care. So that's why I am focused on this. I want to know first all the properties and then how to interact with nanoparticles.
Thank you very much.
Thanks, Gustavo. As with Doug, I didn't know much about mucus. So quick question around, is all mucus the same? So you showed in your rheology that it changes with pH, but is the compositional or the chemical properties of mucus the same in your stomach as it is in your lower intestine?
Thank you for the question. Actually, it's a very interesting question because, well, actually we have a mucus in almost every part of our body. For example, also in our nose, in our mouth, in our lungs, among other places. But the important thing is that each organ has different composition in the mucus. In general, it has like the same elements, but the concentrations are quite different. For example, in the stomach, the main component is mucin. That is the most important protein here. The concentration there is around 5-6%, but in our intestines is a little bit less. It could be around 3-4%. Assuming that the person is healthy, because also during diseases, this concentration can be modified. So also it's important to mention that because as well as the concentration is different, so the behavior is different. Also, it's there are differences in terms of the thickness of the mucus. For example, there are places where we have around 100 nanometers of thickness, like in the small intestine, or in the rectum, for example, there could be even up to 800 nanometers thickness. So it's quite important to consider all these differences. So actually, in the future is one of the next steps. I want to consider all the different stage, well, the different concentrations and the thickness in order to see which area could be the best one for delivery and a specific kind of nanoparticle.
Cheers, Gustavo.
Do you see opportunity for this in terms of lipid delivery systems, or kind of where is it in terms of what types of delivery systems you're thinking about in terms of the application of lipids? Or
Okay, thank you for the question. Actually, lipids delivery systems are a very good ones because mucus is lipophilic, so it's a good one for the penetration. But in my case, I am more focused on polymeric delivery systems because, well, my idea is to have some delivery system that has more specific targeting, and I know more about how to modify the surface of nanoparticles, polymeric nanoparticles, in order to have a targetings, ligands, or different elements in order to specific targeting. But also lipids are a very good one options.
okay, have you taken also into consideration biofilms at all? Because I know that definitely plays into the availability of mucous membranes and things like that.
Sorry, can you repeat your question, please?
Have you considered looking at various biofilms and because that definitely interacts with kind of the mucus availability in different systems?
Okay. Well, actually, until now, I haven't considered the biofilms. I am more focused on, well, to develop or to study these interactions between nanoparticles and mucus with drug delivery idea. But also the biofilms is an interesting topic. I need to read more about it because I only have like the general idea, but it's an interesting topic.
Thank you. Nice job. Thank you.
No, thank you.